So today I will discuss about the enzyme. So introduction part, what is enzyme? Enzyme is nothing but a biological catalyst that catalysis of the life, that catalysis of a different type of biochemical reaction as well as physiological reaction and simultaneously various metabolic processes. Most of the enzymes are generally globular in shape, and it consists of the tertiary and quaternary structure. Now, coming to the point, if I am specifically telling the function, what is the main definition of the enzyme? That is the thing. The enzymes may be defined as the biocatalyst synthesized by the living cell, which they are produced generally in the protein, except protein also it is there, but generally it is a type of protein molecule which is colloidal and thermolayable in nature and specific in their action. Now when I'm concentrating about the non-protein enzyme, so here you can see that the ribozymes, which are the non-protein enzyme, that is a RNA species which act as an enzyme, but that is a non-proteinous in nature. So ribozyme is a type of enzyme where the enzyme is not at all a protein molecule. Next, if I'm concentrating on the importance of the enzyme, enzymes play an important role in metabolism, diagnosis, as well as in therapeutics. So, all biochemical reactions and the enzymes are catalyzed in living organism. Specifically, the various type of biochemical as well as some physiological reaction and the metabolic process are controlled by the enzyme. The enzyme level of the enzyme in the blood are of diagnostic importance. It is a good indicator of various type of disease. If you are having some liver disease, if you are having some kidney function, kidney problem, so if you are measuring the HGPT in case of liver, HGOT, so by measuring this type of enzymatic in levels of this type of enzyme, we can tell that your liver and kidney is working properly or not. So enzyme can be used in therapeutically such as digestive enzyme. When if we are taking lots of food, suppose if it is indigestion যদি আমাদের হয়, then we have to take some enzyme. We have to take some enzyme in different forms we can take. That is the imi enzymes are there. So it functions, it will go to our digestive system, it will go to our stomach and it will digest our food. So enzyme can act as a digestive enzyme, and it is a therapeutic use. It has some therapeutic use. So now if I'm considering about the classification of the enzyme, what do you mean by what is there in that, based on which we can classify? Generally we can classify based on the International Enzyme Commission. So there are major six classes are there. Generally, based on three bases are there. First one is the based on the nature of the substrate on which the enzyme it is acting. Number two is the based on the type of the reaction. Then how the reaction is occurring, how enzyme is reacting with the substrate. So uh, and the last one is the substrate as well as the reaction type, both is involved. So based on that, if you're going to classify the enzymes, so based on the International Union of Biochemistry, so there are major six classes of enzyme. The first class is the oxidoreductase, which is nothing but the oxidation and the reduction reaction. It involves several types of oxidation as well as the reduction reaction. So here you can see the examples is the pyruvate dehydrogenase. We catalyze the oxidation of pyruvate to acetyl coenzyme. So here lactate dehydrogenase is a oxidoreductase enzyme where it acts on the pyruvate and it's converting it to the lactate. Another one is the transferase. Transferase, what is it? It is by name, by seeing the name you can understand it is catalyzing and transferring the chemical groups to one compound to the another compound. So The example is a transaminase, which transfer an amino group from one molecule to the another molecule. So here we can see that the D-glucose, if we are treating with a hexakinase, it converts to the D-glucose 6-phosphate. So hexakinase is such a type of enzyme which is engaged to transfer the phosphate group from the glucose to forming a glucose 6-phosphate. So another next one is the hydrolases. Hydrolases mean it is catalyzes the hydrolysis of a bond. Hydrolysis of a bond is presence of water molecule. So pepsin hydrolysis the peptide bond in a protein, as well as several other enzymes also there, amylase, sucrase, and cellulase. Here sucrase is such a type of enzyme. It is working on the sucrose. Sucrose is converted to the glucose and fructose. So sucrase enzyme is acting on the sucrose molecule, and it is producing glucose and fructose by hydrolysis enzyme. So then after that it is coming lyase. Lyase means the breakage of the bond without any type of catalysis. So here we can see the example is pyruvate to acetaldehyde. Here pyruvate decarboxylase enzyme is working here. So pyruvate is when pyruvate decarboxylase is acting on the pyruvate molecule, it is converting to the acetaldehyde and producing the carbon dioxide. Then after that, one more is coming, that is isomerase. That isomerase, it's catalyzing the formation of an isomer of a compound. For example, various type of phosphate group from one position to the another same compound. In glycogenolysis, the conversion of glycogen to glucose for quick release of energy. So this is a isomer form. So isomerase is such a type of thing, it will just formation, it is helps or it is catalyzes the facilitate the formation of an isomer of a compound. And the last one is a ligase. That means that it catalyzes the joining of the two molecules. So here example is DNA ligase, which is involved in the catalysis of the joining of two fragments of DNA by forming a phosphodiester bond. So for today, that is a thing where we can understand the basic nomenclature and the classification of various types of enzyme and the reaction involved in it, and what are the specific examples involved in it. So basically today this is a thing.